All right, let's go ahead and get started for today. Welcome everyone to our first webinar of our 2024 to 2026 cycle. The leadership cycle for ISBM runs between our in-person meetings. So we just had a wonderful in-person meeting in Toronto last month. And we are excited to, I guess, 2 months ago now, since it's December, we're excited to kick this off for the next season. So for those of you that don't know, the International Society of Bone Morphometry is a society dedicated to all aspects of the quantitative assessment of bone across scales. We have leadership panels and early career opportunities, and we'll be kicking off our working groups early in the next year. So keep an eye out for our communications about that. You can find us on X at ISBM Society or on our website. And so please reach out if you have any questions or want to participate in our society activities. And today I'm very excited to announce our 2024 to 2026 class of the Early Career Investigator Committee. This will be led by Matthias Walla and Ankita Agarwal, our new chair and co-chair, and all of our fantastic members. So they're the ones that are organizing these webinars for you, and they'll be bringing a great slate of speakers forward in the next year and a half. And the other thing I wanted to let you know before I turn it over to Matthias and Ankita is that we are already planning our 2026 meeting. There's going to be a lot of details that still need to come together in the next year, but the plan is to have it in London in late June in 2026. And so we hope to see many of you again in person there, and we're very much looking forward to it. So without further ado, I will turn it over to our early career investigators, and they will kick off this webinar.
Oh, yeah, thank you, Erica. It's a pleasure to introduce our first speaker of the 2024-26 ISBM cycle, Dr. Shatavisha Dasgupta, who is a clinical pathologist with extensive experience in both wet and dry lab work for translational research in oncopathology. And she's a medical doctor who is trained in India. And then she came over to Europe and continued it at the Erasmus MC in Rotterdam, Netherlands, where she also submitted her PhD dissertation on gynecological malignancies and pre-malignancies. Currently, she's a postdoc associate at the Broad Institute of MIT and Harvard, and is an imperative part of the bioimage analysis team led by Beth Simony. And today's webinar is therefore derived from her ongoing work as a biologist. So Dr. Shatavisha, welcome. And we look forward to telling us all about the image analysis workflows, and in particular, the use of the very popular open source software, CellPro-filer. Over to you.
Thanks, Ankita. That was a lovely introduction, and I'm super happy to speak to the ISBM Society. So I would just get started and share my slides. I hope the deck is visible. Cool. So thanks everyone for joining today. Good morning, afternoon, evening, depending on where you are joining from. So today I'll be basically going over the role of quantitative analysis of images and how they can inform various different biological research questions. So historically, analysis of biological images have not been quantitative. So as we go further back in time, we will see that biologists used drawings of cells and tissues to study them. And we don't have to even go that far back in time when biologists were studying cells under light microscope and then using maybe graticules to make measurements. But now, with the advancement of technologies, we have very fancy microscopes, which actually produce images or capture images that are not necessarily very different from the images that we capture on our smartphones, because these are, excuse me, digital images which comprise pixels. And each pixel or a picture element essentially has a numerical value. What we see in the form of an image is just a color representation of that numerical value so that we can appreciate that image as something our eyes and our brain can make sense of. But when you present that image to a computer, it will see it as a huge array of numbers. So you can imagine if you image cells that have been labeled with many different fluorescent dyes and you have many different channels, and if each channel can contain a huge array of numbers, when you combine all these different channels, what huge data set you're getting. And so from this huge data set, if you then perform many different measurements, you want to perform downstream analysis, you can basically end up with a wealth of information that was before not necessarily accessible to the biologist. But any sort of quantitative measurements, we should not attempt to do visually. And why? That's because even for folks who do not have, let's say, color blindness, we vary a lot in how we perceive colors. So this is basically an excerpt from a test that went viral some months before. You can check out the website ismy.blue, and this was actually highlighted in a Guardian article. And you can try this out with your friends or your family members, co-workers, and you'll see what we perceive as green and what we perceive as blue differs widely between individuals. So you can imagine if we try to analyze our research images manually, that can lead us to making very spurious conclusions. That is primarily because our visual interpretation of images can be very much biased by the spatial or the neighborhood information that we see. So for example, on the top panel, the circles in A and B are actually of the same brightness, and the bottom panels, the C and D circles are actually of the same size, but their surrounding objects may make us conclude otherwise. So one question that may arise at this point is, we have so many advanced single cell techniques. Why do we still need images? We have single cell, let's say, many different omics technologies. So what extra additional information can images give us? And that is big. And the great thing about images is when you study the image of a cell or a tissue, you do not dissociate the biological material. And what that can give us is, for example, if you take a look at these images on the top panel, and if you just measure this green marker, in between the images on the left and the right, the mRNA or the protein levels, if you just do a measurement of that, you will see it is the same in both conditions. But if you just take into consideration the localization here on the left, you'll see it's mainly in the cytoplasm, whereas on the right, it is located in the nucleus. And that may have a lot of biological relevance. And if you just use a dissociative method to study these two different conditions or perturbations, whatever the case may be, this context can get lost. So basically, image analysis not just allows us to measure changes in expression of a molecule, but it also helps us actually make more sense of it and understand whether this change in the level is associated with a change in the way in which that molecule behaves inside that cell or tissue. So image analysis is great. I hope I have made that point, but we obviously need computational tools to do it because, as I also showed, trying to do that manually is not a good idea. And what automated image analysis can allow us is make very reproducible measurements. It is a much faster, less tedious process. It helps us measure even those subtle changes that our eyes cannot perceive, and it also quantifies heterogeneity at a single cell level. And so for making these kind of measurements, we need some fantastic open source tools, and CellPro-filer happens to be one of them. So CellPro-filer has been around for quite a bit now, and it is free and open source, as I mentioned. It is very highly popular, and what makes it really accessible is it has a GUI, and a lot of the operations can just be performed on a point-and-click basis. So basically, no coding experience is required. However, if there are individuals or researchers, who have coding experience and want to expand the functionalities of CellPro-filer, because it is an open source software, you can do an installation from source. You can write your own plugins or even add on to the modules. So basically, it is a very flexible tool. But CellPro-filer is obviously not the only one, or not even necessarily, we are not saying it is the most fantastic one. There are other image analysis tools available as well. And as we are making more and more progress in the kind of images that we can acquire, our image analysis needs are getting more and more complex. So what's usually the case is an image analysis pipeline may require the use of multiple tools. And for that, ImageJ or Fiji, which is essentially ImageJ with all the plugins included, is an excellent option as well. It is a very versatile software, but the limitation of Fiji is to perform some more complicated operations. It's very useful to have a knowledge of macro scripting. The other tool that I would also highlight is Elastic. And this is a tool which allows you to perform pixel classification or semantic segmentation. And this can be particularly helpful when analyzing tissue images. And currently, we actually have a plugin called Run Elastic, which allows us to leverage the functionalities of Elastic within CellPro-filer. But the limitation of, again, Elastic is it probably cannot be used to make that detailed measurements as CellPro-filer can perform. So how does CellPro-filer actually work? And it is here that I will be giving you an overview of not just the workflow that we commonly use for CellPro-filer, but the general principles of how any image analysis workflow operates. So this is a graphical representation of a usual image analysis workflow. So we first obviously perform the experiments. We use a microscope to acquire the data, and then we transfer the data to the person who will be doing the image analysis, or it can even be the wet lab biologists themselves. The first step to do would be pre-processing the images so that it will make the next step, which is called object finding, easier. And by these objects, we basically mean whatever it is of interest that we are trying to measure inside that image. For example, if it is, let's say, U2OS osteosarcoma cells, it can be the individual cells that we are trying to measure. If we are studying, let's say, C. elegans, it can be cells of C. elegans, or it can be human tissues. Whatever it is our object of interest within our images. The next step would be to then process all these objects within the software that we are using, whether that be CellPro-filer, Fiji, or whichever software is that you're currently using. And ultimately, what we get out of this are huge spreadsheets, which contain a lot of numerical information. And we would then perform downstream analysis on this extracted data to get answer to our biological question. So just to give an overview of how CellPro-filer looks like or how the modules within CellPro-filer works. If you download, let's say, the latest version of CellPro-filer from the website and launch that application, you will see that a welcome screen pops up, which has some different components like a try example one, which if you click on that, it will just load an example pipeline. And the developers of CellPro-filer have tried to make the software as accessible as possible to the wet lab biologists who, let's say, do not necessarily have a computational expertise or a background in image analysis. And so we tried to provide these examples so that they can have a point to just start building their pipelines. In addition, there are also very helpful links. There's a link to the tutorial page where there are many more advanced options of CellPro-filer pipelines as well, which people can just use to learn more about image analysis and CellPro-filer. And finally, there's also the link to the ImageSC forum, which is an excellent place to ask questions, not just about CellPro-filer, but many other open source software like QPath, Fiji, etc., or even image analysis questions in general. In addition to that, the CellPro-filer interface will have this pipeline panel on the left where all the steps, which we call the modules of a pipeline, will populate. And here we have the images panel where we basically drag and drop the images that we try to analyze. Now, to go into the steps of an example image analysis pipeline. So the first step when we load our images into any software would be then to tell the software how to make sense of these images. So basically, what are these images that I'm providing it to them? And this is important to determine because all the operations will not necessarily performed on every kind of image that we are providing. So let's say I have an image set where there are 3 different channels, blue, red, and green, and blue channel contain all the nucleus staining. So for example, when I will be performing object finding for nucleus, I have to do that on this channel images and not on the red or green channel. And this is why it is important for the software to know what is the blue channel and that it has information on nucleus. So this is done by some modules which are called the input modules, and these are 4 in total in CellPro-filer. And I'll be going over what each module does in the next slides. But essentially what we are trying to do is we are trying to define an image set, which the most common definition of which is where every channel is described to the software. But it can also be configured to mean different things depending on the type of images that we are trying to analyze. So this input modules, as I was mentioning, there are 4 in total. Not all of them are compulsory. So images is obviously compulsory. We have to provide the software images for it to analyze. And the metadata module is actually optional. We use this particularly for high content images from high content screens because these image names often contain information such as the plate number, the well number, or the site, and so on. And we can actually extract all of these information using the metadata module. Names and types is a compulsory module because this is where, as I was showing in the previous slide, we define the image set, or we tell the software that which image belongs to which channel so that we can determine which channel we want to extract what information from. And groups is a module we particularly use only for Z projection or sometimes for 3D experiments and so on. So this is not a mandatory module either. And so all these 4 modules, we have to keep in mind that they act in concert. So we have to make sure that they are all configured correctly, or the downstream processes or the other downstream processes will not be able to take place. So just to give an example, for example, the metadata extraction, as I was mentioning in the input module, it extracts information from file or folder names, but it can also import this information from an outside file like a CSV file, if that is how you have saved your image information. But quite often from high content screen, as I was mentioning, they contain the well position and the site or the channel number within the image file names. And we can actually configure some regular expressions like these. These are following the rules of Python. And we can explain to the software that which part of the file name, what information belongs to. So which part means a plate, which part means a well, site, and so on. And this is important because at the final step of all the of the pipeline, when we will be exporting the data to spreadsheet, all this information will be getting attached to that spreadsheet. So we will know exactly from which well, which site all this information is coming from. So, and then the next thing, as I was mentioning, is the names and types module. And this is where, again, we can set certain rules. So for example, let's say the image name, as we can see here, for example, has CH4, which is basically indicating the channel number. So we can, and here, for example, let's say there were additional ones which had CH1, and we can indicate that if there's a CH1 in the file name, please assign this particular name, and you can set whatever name that's appropriate for your experiment. So once the software has an idea of the kind of images that has been provided to it, the next step would be to find our objects of interest, or whatever that is we are trying to study within these images. And this is known as segmentation. What we perform within CellPro-filer is not semantic segmentation. It is basically using the traditional OpenCV methods, and we do thresholding for most of these purposes. And that is, and that's based on the basic premise that my object of interest within an image is the brightest thing. and everything else in the image is darker than my object of interest. So to make this work, what we do before performing segmentation is sometimes we do some pre-processing. So that may need some enhancing of the signal or some blurring, depending on whatever kind of image that we are trying to analyze. And this is all these operations are essentially done to optimize the signal to noise ratio so that we can identify our object of interest in the best manner possible. But we have to keep in mind that all these pre-processing steps that we do, these are only for identifying objects. The pre-processed images are never to be used for making any kind of measurements, because we have, let's say, enhanced a signal or blurred an image, any kind of measurement that we perform on these will be giving us absolutely spurious values. So just for segmentation purposes, any transformation is legal, but that's not necessarily true for making measurements. So what kind of pre-processing can we do? So there are many options within CellPro-filer. We can enhance edges, we can enhance or suppress certain features, smooth, or there are even more advanced options like applying Gaussian filter, remove holes, etc. All these are quite well documented within the CellPro-filer manual, and there are also some example tutorials that I would really invite anyone who is more interested to learn about this to check them out. And again, these are chosen depending on the kind of images that we have at hand. But if we just break down the segmentation step, it essentially comprises distinguishing the foreground from background, or the thresholding, as I was mentioning before. And the next step would be, once this foreground is identified, determining whether the foreground contain one object or multiple objects, or let's just say how many objects are present within this foreground. So for the foreground to background, making this distinction, we use a method that's called thresholding. And this can also be determined in a trial and error method within what the ideal threshold is can be determined in a trial and error fashion within CellPro-filer. And we have to be cautious about that, because as we can see, if we set the threshold too low, we can include some pixels which do not necessarily belong to the object of interest. And if we set it too high, we may lose some important pixels that belong to the object. And we just have to figure out what's just the right spot or the right threshold. And for that, this is how the identify primary object, which is one of the modules that we use for segmentation, look like in CellPro-filer. And within this drop down of thresholding methods, we can see that there are many different options available, and we just choose the one that's most suitable for the image. And once we have performed this identify primary object, this is how the output would look like. And as you can see that there are many different colored outlines around these objects. So in this case, the nuclei have been identified, and the colored outline basically mean what's the interpretation of the CellPro-filer about these objects. So green outline means the objects that have been accepted or which fulfill all the parameters that we have inputted within the software. If some objects have been identified, but they do not fulfill the parameters that we have supplied, which I think in this case is the size, because I have set a minimum size and these have been identified, but they do not fulfill the minimum size. So the software is rejecting these, and these objects are outlined in magenta. And there are some objects which are on the rim, and these are outlined in orange. And these are usually the objects which touch image borders. And these are objects which we usually discard in an image analysis workflow, because usually the edges of the images often contain a lot of artifacts, but this is completely optional, and we can just set the button, reject object touching image borders, to know if we want to include these objects in our measurements as well. And of course, we get a summary table of the number, diameter, and some important measurements within the output pane of this identify primary object module. So as I was mentioning, once we have identified what's foreground and what's background, we would want to figure out how many objects are there in the foreground. And this involves a very important step, which is separating the touching objects, or separating all the different objects that we have identified in the foreground. And the principle that's used in CellPro-filer is called a watershed method. This also comes from OpenCV concept. And here, basically, each image is considered to have a peak intensity, which also signifies the peak of a mountain. And in between images where the intensity progressively dims, these dimmed areas are considered valleys. So what the computer essentially does is then it starts filling out the valleys between these intensity peaks with different colored water. And at the point where these water will be starting to merge with each other, it will start creating barriers. And these barriers are essentially what are identified as an object boundary. And so it will continue doing that or filling these valleys till all the peaks get submerged underwater. And that's how we end up having the outlines between the touching objects. And so there are also some options of determining how to do this in CellPro-filer. We can set whether we can use the intensity levels to demarcate the object boundaries, or we can use shape. And this again is defined by the kind of object that we have in our images. For example, for let's say the rounder object, we can use the option shape to separate the touching objects. And the other parameter that makes a lot of difference in separating touching object is defining the local maxima. So these intensity peaks that I was mentioning, that each object has one intensity peak. So these peaks are actually called the maxima. And so the distance between these two peaks or of two neighboring objects will actually determine whether it is to be considered as two distant objects or one object. So if we set the maxima distance to be too low, it will be splitting one object into two. Or if we increase it, it will then actually correctly it is correctly identifying the object as one in this case. But again, if we set it to be too high, and if we have objects kind of touching each other, it may also clump it. So this is a parameter that needs a bit of tweaking to make sure we are actually identifying the neighboring objects accurately. So this is how the interface would look like. For example, here you can set the method to distinguish clumped object as either shape or intensity, as I mentioned, and also we can try out different numbers for the local maxima to determine what's giving us the correct segmentation. And this is an example from CellPro-filer. So what happens when we set the local maxima to be too low? We can see that this is actually one object, but because the local maxima is set too low, it's incorrectly sort of over-segmenting it. But if you just perform some trial and error, you can figure out the correct number. And here we can see that it is performing perfectly. So once we have identified the primary object, and which in almost every case is the nucleus if there's a nucleus stain available, and the reason behind that is nucleus stain is usually the one that is least variable and ends up being the most easily identified. So we usually start with segmenting the nuclei as the primary object. The next step would be then identifying secondary and tertiary objects. So secondary object would be, in case of CellPro-filer, the cell. And for that, we can use either the cytoplasmic channel or the cell membrane channel that we have. And there are also, again, several options like distance n, which does not even require a stain to be present in the membrane or in the cytoplasm. Or there are other methods as like distance B watershed, which utilizes the staining intensity in the cytoplasm or in the membrane. But all of these methods would be using the primary object or the nucleus as a seed, and it will start expanding pixels from this central seed to define these secondary objects. And as I was mentioning for distance n, what it does is, because this is usually the method we go for when there is no cytoplasmic or membrane staining, we just assign a specific number of pixels by which the nucleus boundaries will be expanded. And let's say if it's 10, it will just expand the nucleus boundaries by additional 10 pixels, and that boundary would be then considered as a cell. And tertiary object would be just nothing but subtracting the nucleus from the whole cell, and that is how we end up with the cytoplasm. And the point of doing all of these steps is to basically break down each object that we have identified, and this object meaning a whole cell, into different compartments, because next what we will do is perform measurement of these different markers that we have present in the nucleus or in the cytoplasm or in the whole cell, so that we can then perform even more further downstream analysis and figure out our biological research question. So in addition to these, just identifying the cell, cytoplasm, or the nuclei, so what if I want to identify an object which lies within an object? And one example of this would be, let's say I have a marker that's expressing the RNA speckles, and I want to quantify these speckles, but I just want to quantify the speckles that lie within the nucleus. So, for example, this is the image from an RNA channel, and we can see that there are some of these dots which are present within the nucleus. So there's actually a module called Enhance or Suppress Features in CellPro-filer, by means of which we can just make these dots or speckles a little brighter. We can first identify the nucleus using the Identify Primary Object module, and then we can apply the nuclear masks, so that we are telling this CellPro-filer that please identify in the next step the dots that are present within this nuclear mask that I have applied. And so I can do another round of Identify Primary Object, but then only in the areas within the nuclear mask. And as you can see, I have now accurately identified all these dots that were present in the nucleus. And this is essentially what we will end up with, where the red boundaries are showing the nucleus outlines, and these dots are showing these RNA speckles. And if it is important for me for this research, let's say I can make a count of how many speckles are present in in which nucleus, and then I can make a comparison across different conditions or time points. So once objects are identified, the next step would be to measure everything. And this is a principle that our lab or the imaging platform goes by. So we try, the motto we live by is measure everything and ask questions later. Because quite often when we start with a research question, we think, oh, this may be the parameter within the cell that will change when we have introduced this perturbation. But what we see is within images, there is actually so much wealth of information available, and this automated measurement, automated analysis makes quantifying everything so easy. It is best to measure everything, because quite often we are actually surprised to find out that the aspect of the cell or the phenotype that ends up getting changed or being most informative is not necessarily what we thought would be the most valuable one. And there are many different modules in CellPro-filer to do that. There's measure object size shape, which can give you measurement of area, perimeter, etc. Object intensity, which not just measures the total mean, median, or standard deviation. In addition to that, it can also do a measurement of the intensity distribution, where it can actually start drawing concentric circle from the object center. And for each of these concentric circle, it can give you an intensity distribution of the marker that is being expressed within that object. And this is very, very informative and can allow performing many really fine-tuned downstream analysis. And another important measurement that we do is co-localization. And there are many different options or measurements that CellPro-filer performs. So co-localization can actually mean, and that's because co-localization can mean different things. It can mean correlation or co-expression. So let's say I have a marker that's tagged with GFP here, another marker that's tagged with mCherry, and I want to find out whether the same parts express both of these markers. And co-expression or correlation, as I mentioned, that they can mean many different things. For example, two markers can be expressed in the same place, but there may not necessarily be a correlation with one marker increasing and the other marker following suit, whereas the markers may not be, for example, here, they may not be expressed in the same place, but they may actually have a strong correlation of when marker 1 goes up, marker 2 goes up as well. And so all of these different measurements we can again perform within CellPro-filer. And these are the other additional measurements. There's, all the names are pretty self-explanatory, so I'm not really going into a lot of depth in these. So you can measure the number of neighbors, and you can also fine-tune to determine like what would be the distance or the boundary within which the neighbors are to be counted as well. And finally, once all the work is done, it would be important to export all the measurements, and we can export that to either a spreadsheet or to a database for use in further classifiers. And we can also save the project as a CP-proj file, which will contain the image locations as well. Or if we want just to save the pipeline, we can save and export it as a CP-pipe file, which is actually very portable and can also be submitted as a part of our publication, because reproducible research is something we strongly believe in, and it will allow another researcher to actually replicate all the analysis that you have done with your images. So in addition to these modules, CellPro-filer also has the option of using plugins, and that allows you to get more out of CellPro-filer. And why do we need that? That's because segmentation, as I was, I think I talked quite a lot about that, is actually a very difficult thing. And that's what image analysts struggle with quite a lot. So on top, this is the results from a survey conducted in 2020, where you can see when image analysts were asked, like, what's the challenge that they encounter, the highest response was segmentation. And this can be particularly challenging when you have elongated objects like bacteria like this, donut-shaped cells, or if the nuclei are crowded and if the nuclei are of many different sizes and shapes. And so these are challenges that CellPro-filer can tackle, but sometimes it requires a lot of manipulation. But particularly in cases where there's a lot of overlapping or crowding, or when there's a huge variation in the sizes of the nuclei that you're trying to analyze, deep learning can actually be a very useful option. So recently, we have had many different deep learning algorithms that have come in. For example, CellPose is a fantastic one. And it is a generalist algorithm where it basically uses manually annotated masks and transforms them into vector flow representations. And these vector flow representation can actually be then interpreted by a unit, which is the structure, neural network structure on which CellPose is built on. And what it then does, it starts creating these simulated diffusion, the simulated diffusion representation from which it derives spatial gradients. And it combines the gradients from the X and Y axis to between 0 to 360 degree. So you end up getting these flow threshold and probability threshold like these. For example, here, it's represented how these combined gradients would look like. And these are then used to generate object masks. And they work very well for crowded objects or like really challenging objects, which CellPro-filer can struggle with. So we have a plugin called Run CellPose, which allows us to use CellPose within CellPro-filer. And these plugins, we have made sure to make them available not just for CellPro-filer that's installed from source, but also that's actually pre-built and downloaded from the software. And these plugins are usually the ones which tend to be under active development, and these are not included as modules within CellPro-filer because this can be catering to maybe a more niche audience. But with Docker or this containerization, it is actually possible to use it's actually accessible to biologists who do not want to use a command line, and all of this can be done completely on a point-and-click basis. So what people just need to do is to have, let's say, a Docker desktop application on their computer. They need to pull the images in which the plugins reside. And they would need to, when they open up CellPro-filer, just point it to the image that needs to be, the Docker image that needs to be operated. And then you can absolutely use CellPose within CellPro-filer. You can use any of the pre-built models, or even if you have done some model training with CellPose, you can also point it to the model file within your computer, and you should be able to utilize CellPose within CellPro-filer for your more advanced segmentation needs. And this is just to give an example of things that CellPro-filer tends to struggle with. Here, like, there's quite a bit of variation in image size and shapes, and this is what CellPro-filer identified. But as you can see, like with CellPose, we are just getting a bit more accurate boundaries. Last but not the least, and this is, a very extensive part, but I will just go over in very minor details, and I can answer questions. So how does all these measurements in this, can essentially drive biological discovery, or how does this actually help the researcher? And you can essentially use CellPro-filer with any image from any experiment, but so far it has been quite extensively used for high content screens. So what people do for high content screens is use a staining protocol like cell painting, but essentially any other staining protocol can be used. The principle that it follows is just try to label as many compartments of the cell as possible. So in cell painting, we have 6 stains or 5 channels, and it reveals 8 different cell compartments or organelles. Supply all of these images in the manner or in the sort of the skeleton pipeline that I just described to CellPro-filer, make every kind of measurement, and then do downstream analysis to figure out the effect of the perturbation or the experiment that you have subjected your cells to. So people tried to do that with cells that have been subjected to, let's say, genetic perturbation, many different types of chemical perturbation for drug screens, and so on. And what they essentially how they essentially try to utilize this data is figure out the phenotypes of the cells subjected to many different conditions. And based on the similarity or the dissimilarity of the phenotypes of these cells, which is interpreted based on, again, the measurements that was performed on these cells, utilize that to determine whether the conditions that they were subjected to are similar or dissimilar from each other. And it was actually noticed that just by using these CellPro-filer features, we can actually predict the functions of many different compounds or many different classes of compounds with a very high percentage of accuracy. And this whole methodology, it is quite gaining, it sort of came into prominence about, let's say, a decade back, and it is quite gaining in popularity now. This whole methodology is called image-based profiling, and it is a very low-cost method which can be scaled to medium and high throughput experiments. And compared to what, say, other genomic or transcriptomic experiments cost, like this is a very, very cheap method, and this only costs a fraction of other omics experiments. And so with this, you can try to predict a mechanism of action of newer compounds. You can identify concentration of compounds that can produce off-target effects. You can also identify new compounds and also cluster compounds into the ones that have a similar mechanism of action. So basically, all of these methodologies combined together can really facilitate drug discovery. And this is how a general image-based profiling workflow will look like. The first step is feature extraction, which is basically making all the measurements with CellPro-filer or any other image analysis software, pre-processing the data, and downstream analysis, like making correlation and visualizing correlation patterns. So as I was mentioning, feature extraction would be using all these modules that I just spoke about, using CellPro-filer or your favorite image analysis software, and again, try to measure as much as possible. Next, in the context of high content screens, we try to collapse the data a little bit, because otherwise, if we have data on a single cell, level, this ends up being a very huge matrix. And sometimes this, a lot of the features end up, those which have, let's say, additive effects. So that does not end up producing very meaningful results. So we try to collapse them into making per well means, so that collapsed data can then, or this collapsed matrix can then be used to compute correlation. We normalize the data to negative controls if there are negative controls present on the well, or we can also do a whole plate normalization. And then we remove the redundant feature. As I said, like some feature tend to have an additive effect, so that we can use a much more collapsed data set for then making Pearson correlation. And this is where we try to figure out which cells are looking more similar to each other. And this Pearson correlation can actually, this data can actually be visualized in the form of a similarity matrix. And with this similarity matrix, we can see whether, let's say, well A01, which is perturbed with compound 1, is very similar to well P03, which was perturbed with compound 3. And so we can infer that maybe compound 1 and compound 3 are actually quite similar to each other. So before ending, I would again just point out, so all the data analysis, image analysis, and any type of quantitative analysis, all of this sound very fantastic, but at the end of the day, it very much depends on segmentation, because segmentation is a step that is determining how many cells we are identifying, how many cells we are counting, and over or under counting can really muddy the waters. And also, sometimes, which does not sound fancy at all, but just looking at the data is very important, beyond utilizing many different advanced deep learning or other methodologies. So on the bottom is a Datasaurus dozen, which basically shows that with the same distribution of the data, depending on how you are plotting it, you can get from a dinosaur to a star to many different visualizations. So it is very important to do sanity checks, to just maybe doing a visual inspection of the segmentation results that you're getting, to also keep in mind the research question that you're trying to address before drawing conclusion from your data analysis. So I would just summarize by saying that I hope I have been able to convince people that imaging data can provide many different answers to your research questions, and we have now excellent tools available which can make image analysis not tedious and rather a many fun process. If you do not have computational expertise, you can obviously use the options that are already available. Or if you like experimenting and creating your own module, open source softwares are an excellent options for that. And I just gave an example of how you can collapse, normalize, and use all the features that you extract using CellPro-filer from high content screen, from driving drug discovery or repurposing drugs. I have also linked some resources here. I can share the slide deck after the presentation, and would also point out anyone who's curious to know more about images to the forum, ImageSC forum. And finally, would thank our imaging platform, in particular Simini Lab, and all the members of Simini Lab. And these are some of the other tools that our lab is currently working on. And happy to take any questions.
Thank you, Shatavisha, for this really nice presentation. I encourage everyone to type their questions in the chat, or just raise your hand and unmute yourself if you have any questions. And maybe I can go ahead with the first one. So for me, it's always really interesting to see, because I'm not in this kind of like microscopy space. I come more from like CT processing and so on. So it's really nice to see kind of like these parallels, but also like these slight differences between these kind of two fields in terms of image processing. And maybe something you kind of touched on in the end as well, but maybe you could give some more detail on this, kind of like the image artifacts that you have to deal with, because I'm sure there's like also some kind of like problems in microscopy images that often occur, and maybe you can comment on maybe how to automatically quantify these artifacts that would kind of like influence your analysis, either automatically or even visually.
That's an excellent question. And I would say not finding artifacts is rarer than finding artifacts. Almost every experiment end up having artifacts. In most cases, if it is, let's say, some debris that you have in these images, and they tend to be, let's say, like very bright spots, so you can just use thresholding to get rid of those, and that makes, depending on how problematic your artifacts are, it is removing them is, can be challenging or not challenging, depending on, like, again, how difficult they are, but. If it is a very uniform artifact, it can be automated, like their removal can be automated pretty easily. What we do very commonly for high content imaging is do an illumination correction, which is what we usually use as a flat field correction, but you can also do some sort of like more extensive background subtraction to make sure you're getting rid of all the noise. But if it is, again, like very, very challenging and the artifact like vary from image to image, that is where like deep learning methods can be really helpful.
Awesome. Thank you. I saw there is a couple of questions in the chat. I don't know what's easier for you, if you want to read them and briefly summarize and repeat.
Yeah. Can the users fine-tune pre-trained model, the plugin? Yeah. So you cannot fine-tune it within CellPro-filer. For that, you will have to install CellPos. But once you have fine-tuned the model and saved it in a location on your computer, when you're using CellPro-filer, you can just point it to the location of that model, and you should be still able to use that trained model within the plugin. The next question is, how does one access CellPro-filer? Yeah, exactly. It's linked in the next comment below. And there, 4.2.8 is the latest version, but we are working on CellPro-filer 5, so we'll keep an eye out on that. Computational requirement of the workstation to run. It can be on any workstation to run the analysis. It rather depends on how big is your batch. So if it is, let's say, from high content screen and you have 50 plates and many hundreds and thousands or close to 1 terabyte of images, you would want to do that on the cloud. We tend to use Amazon Web Services for this purpose, but again, it can be run on any cloud. Also, we know some labs which run, let's say, or smaller batches on high performance clusters. And it is also possible to run CellPro-filer on a headless mode. Is the workflow only 2D images? No, it works on 3D images as well. Whole mount images with many Z-stacks. I have not personally worked with bone images, but I would really encourage you to check out the 3D tutorial that we have. So we have used 3D pipelines for analyzing, let's say, blastoids and other worm images. So I'm pretty sure you can modify that for your needs. Files can be moved across software. Yes, you can move our files, but Imaris or Amira, I'm not familiar with, but Imaris is a proprietary software. So I'm not entirely sure how you would utilize the CellPro-filer pipeline within Imaris, because the files that you export out of Imaris are usually, they have proprietary file formats, if I'm understanding correctly. So you have to make sure, let's say, if the images that you're exporting out, they are in a TIFF format or a PNG, and then you should be able to use that within CellPro-filer. Histological sections. Yes, we can also use CellPro-filer. I have actually worked on projects on tissues as well. Not bone tissues, though. These were cancerous tissues from other organs. What you would essentially need to do with the current version of CellPro-filer that we have is whole slide images, because they are so huge. You cannot load whole slide images in CellPro-filer in an untiled format. What you would need to use is, let's say, either using QPath or a Jupyter notebook, create tiles out of your images. and feed CellPro-filer these tiles. And then essentially the workflow that I mentioned is the same one that you would use for your tissue images as well. And then let's say you want to quantify some marker expression within your cells, you would then use additional thresholding steps to do your cell positivity calling. But in CellPro-filer, this may change in CellPro-filer 5, and we may be able to load even larger images in CP5. CellPro-filer having issues with an image data size. Yeah, that's what I was just talking about. Large multiplex images problematic on slide. Yes, so we are working on that. Hopefully that will be solved. But as I was mentioning, so I have actually worked with multiplex IF images on CellPro-filer, and what I do is I use a small script written in Python to make these tiles. And these tiles actually contain the coordinate information as well. So I can make all the kind of measurements in CellPro-filer, and using these coordinates, I can then map them back to which part of the slide it came from. So essentially, just the skeleton pipeline that I just went through, you can use that in CellPro-filer as well. But yes, at this point, you would need to tile it up first. Negative staining needed to optimize the workflow. We actually have an excellent blog post for good enough segmentation. So I can add that in the slide deck, and I'll share that with the participants. Negative control staining for the workflow. We don't usually incorporate or ask our collaborators to incorporate any negative stainings necessarily, but for normalization step is usually where we use the negative controls. How to apply principles and tools, imaging at other. You discussed imaging at other lens scales. I'm not sure I understand the question very well. If you mean like scaling it up in terms of the image sizes that you have, I think in that case, you would have to make sure you have the infrastructure for the compute. If that is what you mean, I mean, you're very much welcome to unmute and ask me. But as I mentioned, like for the larger batches, we tend to not do these analysis locally, but we do that on the cloud on Amazon platform, or larger imaging structures. Okay, bigger than cells. Yeah, so if it is like tissue images, again, or like a larger image file size, so in that case, again, like tiling them up would be important. So that's what we currently do for all tissue images. And again, if one is computationally comfortable, it's possible to do that with a Python script, or it's actually quite easy to do in QPath as well. And that does not necessarily like need any scripting. And these these tiles can then be used in it in CellPro-filer. Yeah, I hope that answered the question. I think setup capacity. Oh, that's such an excellent question. Yes, we can identify multinucleated cells, but you need the plugin run CellPose for that. And I'll tell you why. This is not something I touched upon. And that's because in CellPro-filer, what it does is when it identifies a nucleus as a primary object, it identifies the whole cell as a secondary object, right? And it enforces a one-on-one relationship with the nucleus and the cell. So even if there's like multiple nuclei within one cell, it will sort of still enforces one-on-one relationship. So what you would essentially then need to do is use run CellPose. And that's because like that's how conventional machine learning works. So what you can do is use run CellPose to identify the nucleus in all the nuclei in one step, and then you can identify all the cells again using run CellPose in the next step. And then you can use an object, a module called relate objects in CellPro-filer. And what it does is it takes one object and it finds how many objects are present within this object area. So it will then assign within one cell area how many nuclei are there, and it will be assigning them to that cell object. And so essentially, at the end, what you get is a count of your multinucleated cells. Yeah, I hope that made sense. But yeah, that would be the way to do it.
Thank you. I think, yeah, we're at the end of time.
Yeah.
But yeah, thank you for taking the time to present today. And it was a really interesting presentation. And I hope to see many of the participants at our next webinar next month.
Thank you so much.
Thanks for
Bye.
having me. Bye.
Thank you.